Hi guys, quick lecture today on DNA replication. So this is still in your 2.6 notes, but it's like the last couple of pages. Okay, so if you'll scroll down, it's still in this 2.6 notes. So open up that notes document, and this won't take very long. I kind of explained it the other day. So number one, why does DNA have to have a way to replicate itself? This ties into cell division. Remember when we said you're going to have one cell dividing into two cells, okay? In mitosis, if you remember, that was mitosis. You have to copy the DNA or copy the chromosomes, which is where the DNA is. So now we're looking at when we talked about, how a chromosome makes a copy of itself and it's attached as a sister chromatid. We're looking at the process. Now that we know what this is made of, if I were to look up close in that chromosome, I would see DNA with my A's, T's, G's, and C's. Okay, so we're looking at up close how we make a copy of that. So if you need to pause, you can pause and fill this in. Why do we need it to replicate? So that when a cell divides, each daughter cell gets a full copy of the genetic code or the DNA. The genetic code is the DNA. Number two, when does that occur? Just to remind you, that happens during the S phase of interphase. Remember, we said that's when DNA replication occurs. Here's an overview of the process. Before we don't really have many notes to take on this. It's really easy. If this is a DNA strand, okay, and remember, we have our A's, T's, G's, and C's. I'm going to do, we have our hydrogen bonds in here, which I said were weak bonds, okay? All right, I don't feel like doing them all, but anyway, those are all my hydrogen bonds. If you remember, we said those were weak bonds, which means they can be broken, okay? And we're going to see here, this is when we want to break them. And so it's going to be sort of like we say it's like a zipper. We're going to unzip this. If I want to make a copy of this molecule, what I do is unzip this, or in other words, break the hydrogen bonds and separate these two strands. And you can see that happening here. And then we have these exposed bases, and we use the complementary base pairing rules. An enzyme's going to do all of this. And there's going to be these free nucleotides hanging out in your cell, in your nucleus. And remember, there's 4 different kinds, depending on whether it has A, G, C, or T. And you just use the base pairing rules. If there's a G, you put a C. If there's a T, you put an A. And an enzyme does this. And you should be able to do this. If I were to give you, this strand, you should be able to tell me, for example, what would the next thing be? If it reads a C, what's it going to put right there? It's going to put a G nucleotide, okay? And then I'm not going to draw the whole backbone. If there's an A, it puts a T. If there's a G, it puts a C. If there's a T, it puts an A, okay? And so you just keep using the TA, AT, GC, CG base pairing rules, and you do that on both sides. An enzyme would do this, but you should be able to tell me. Anyway, and do that all the way up. And using the base pairing rules, that's essentially how it copies. So by copying complementary base pairing to this left side, you're essentially getting what was on the right side. And same thing here. By reading the right side and using the base pairing rules, you're essentially getting what was on the left side. And then this, so this is sort of in a process of how you get it. Then you end up with this. Okay, so number 1 says label the parent strands and the daughter strands. So the parent strands are the ones that we start with. Okay, so these are the parent strands. I'll label those in blue. That we're starting with. Okay, and again, you can see there's the parent strand, there's the parent strand, and then once we're done, each DNA molecule has one original parent strand, and then the daughter strands, I'll put in red. The daughter strands are the new ones that were made, just like when we did cell division. Daughter was the new cells. So those are the new daughter strands, okay, the ones that were just made. Okay. Sometimes we refer to the parent strand as the original strands that were on the sides of the DNA ladder. Okay. So that's letter A. We labeled those. What are the complementary base pairing rules? Okay, well, we already know A goes with T and G goes with C. And then it says, notice the DNA molecules have the same, this is really important right here. Notice the two DNA molecules formed have the exact same base pair sequence as each other. Okay, so if you read these two strands, and you read like down or up, they have the same thing. A, T, T, A. A, T, T, A. C, G, G, C, C, G. This order of bases is the exact same as that. And that is also the same as the original DNA molecule. So if I go back and I read, okay, so let me just highlight the original DNA strand, and then the two that I now get after I make a copy have the exact same DNA sequence. And that's the whole point. You don't want to copy the DNA and make a mistake. That's a mutation. Okay. And then a lot of people often ask, where do these nucleotides, remember these are nucleotides. Remember, a nucleotide is a sugar and a phosphate and a base, like A. But why is it doing that? It won't let me write. It wants me to. All right, anyway, A. So that's like a nucleotide, sugar, phosphate, base. So that's what this is simplifying. So these individual nucleotides are floating around, and they're there as building blocks to add on when our DNA replicates. Where do they come from? From the food you eat, okay? So let's say you eat a steak, right? What are you eating? You're eating cells of a cow, and those cells have a nucleus with chromosomes and DNA inside. And when you do digestion, you take those cow DNA in their cells and chop them up into individual nucleotides. That's happening during digestion. And then that gets delivered by your blood to all of your cells. And then we're going to use those as the building blocks to build our own DNA molecules. Okay. All right. So that is essentially how it happens. We open it. We base pair to both sides using the rules, and we get two identical strands and just some details. Of course, we just have to make this hard. It's the way that it does that. We call that semi-conservative replication, which means no DNA molecule is ever completely new. Again, you can pause and write this down. And there's always one original strand, which we call the parent strand back here. Original and parent strand are the same thing. And one new strand, okay? The new strand we called the daughter strand sometimes. Okay, so you can see here in this picture, if the two white strands were your original, we opened it up and copied it, that the two strands you're going to get have one original side of the double helix and then one new side. Same thing with the other one. And that's what we call the semi-conservative model, which is what we believe to be the correct model. These are three models of DNA replication once considered. Okay, ... We now know, or we now think, right? Science is always changing. Sometimes we think we know. We now think that that is the model, that is the way it actually occurs. And that's what I just kind of described to you. You open up the original, and then the two you end up with have one original side and one new side for using the base pair. These other methods, conservative, that was a model at one time where maybe somehow the both original strands would end up together and both new strands would end up together. I can't think of how that would happen, but it doesn't happen that way, so it doesn't matter. And dispersive was the other model. Maybe somehow each strand ends up with pieces of old and new. Again, those would be difficult to figure out the mechanism of, and we don't believe it happens that way anyway. So just know this term, semi-conservative, and be able to pick this picture out as the correct mode. And then all we have left to do is just talk about the names of the enzymes that do these things. There's two enzymes, okay? And so the first one is called helicase, And that's the one that's going to, we say, let's put unzip in quotes. It unzips the DNA by breaking the hydrogen bonds between the bases. So again, this picture it kind of left out. Let's draw in the hydrogen bonds. They go right there. All right. And helicase is going kind of in this direction. Okay. And it's going down and just breaking those weak hydrogen bonds. And as it breaks those, it opens up the strand. Okay. So that's helicase. Helicase opens it. And then you can see here in yellow, they're calling it DNA polymerase 3. There's different kinds of DNA polymerase. You don't have to get into it. I just want you guys to know that the enzyme DNA polymerase is the one that then copies it, and it links the new nucleotides on using the original one as we often call it the template strand. Again, it's using the base pairing rules. Okay. And you can see that you're going to have one on each strand then. That's going to be reading down and adding on the proper bases. So I might ask you to do, act as DNA polymerase. Let's suppose I gave you, if I said, okay, here's the DNA template strand, A, G, C, T, T, A, G, C, whatever. And I might say, if you're DNA polymerase and you're copying it, what would the other strand be? And you should be able to use the base pairing rules and put, a T wherever there's an A. And remember, G and C always go together. Whatever. Okay. You should be able to, just using the base pairing rules, tell me what the other side is. All right. The last thing then, we just have you, I don't know why we have you write it again, but there's what DNA polymerase and helicase do. Again, you can pause and just kind of fill that in. Helicase breaks the hydrogen bond between the bases to open up the double helix, and polymerase reads it and adds the proper nucleotides using base pairs. There's a link that shows it all in action. If you want to click on it, I'm not sure it'll work, but and that's it for today. Go ahead and upload those notes to Google Classroom.